Okay,
Oh,
hello.
YouTube.
Hello, YouTube. Okay, hello, class. We're gonna do. This is chapter 8. I know we haven't done chapter 7, but it goes chapter 8, 7. It's weird. But we're gonna deal with photosynthesis. Photosynthesis, I think a lot of you already know what the basics are. But really, this picture sums it up. You need carbon dioxide and water, and then the light energy, and that will make oxygen and sugar. Sugar is what they use. Oxygen is a waste product. I see the light. So we're gonna first talk about pigments. Pigments is how electrons can absorb energy, and what it means by being excited is higher energy level. It has more energy. So chloroplast uses sunlight to make energy, and pigments absorb light of certain wavelengths. So
It's also the color that absorbs the light.
Pigments can protect cells from UV damage. That's a fact out there. Most common pigments reflect green, so it gives color and absorbs light. So here's a picture of wavelengths. Let's say a pigment looks at blue, a pigment will detect red. This is visible light. We all know microwave waves are very dangerous.
And the most effective wavelength is, of course,
Gamma rays. Those are the safest. Gamma rays are the safest ray.
Well, gamma rays is the safest, but more importantly for a plant, the color of the wavelength color would be green, as it's one of the most effective ones.
I think it's blue and red. Remember the lab we did?
Yeah, it's between, they're like at opposite ends. It's like, it's in the green area and then over at like the bluish, violet-like color, I think.
Okay.
If I remember right.
Yeah. Okay, the leaf. We're gonna now go into the leaf because... Okay, so the leaf, we have like a skin, like kind of like our skin. And below that we have chloroplast, which is what happens. We have a vacuole. This is like individual cells. We'll have a vacuole. This is where everything is stored. A nucleus, DNA is stored. Cell wall, sturdy. And cytoplasm, liquid stuff. So here's a very important organ. It's called the stomata. And it's holes that allow... They're like mouths. They allow CO2 to enter and oxygen to exit. They can close, they can open. We'll discuss their closing and opening later. Chloroplast is where photosynthesis happens. Chlorophyll is the pigment. It's the pigment that absorbs light. Stroma is the thick fluid filling the inner components of the chloroplast. It's a fluid, kind of like the cytoplasm.
Ooh, okay. Try not to get mixed with stomata and stroma.
Exactly. I really like
okay.
why.
I think that's going to be stromata. Okay, stromata is the thick fluid. Stomata.
What?
Stomata is the pores down.
Yeah.
Air
Holes.
going through.
Yes. Do they all both have... Okay. How can we, like, differentiate?
Well, stroma has the R in it, so it's stro, but stomata. So I guess,
I think you can hear the O more.
So stomata, I guess, yeah, you can hear the O for oxygen.
O has, like, a hole in it.
Then again, O is just like a freaking vowel. So, I guess,
You can hear the O more in stomata, so maybe whole O.
I look at the T as like the branch with the leaf, so therefore it's on the leaf. It's the leaf part.
They're both on the leaf.
I don't know.
It's okay.
Well, stroma is a fluid inside the chloroplast, which the chloroplast is part of a leaf cell.
Stroma is like a chloroplast. It's just filling the inner components. It's the same job the cytoplasm.
Yeah, cytoplasm.
It's the same. It is the same thing as cytoplasm, but instead of the entire cell, it's chloroplast. Thylakoids, they're sacs, and chlorophyll molecules.
Those are this shape. There's a this shape.
Yeah, it's
parts of the chloroplast.
It's like right here. Can you like see my mouse or?
I'm doing like a copy on my screen, but.
Okay, so so we're in this picture. We describes it. It's really good. So we have two light reactions and light independent reactions. Light dependent and light independent. So the light dependent requires light. The light independent does not. So the light independent is another name for it is the Calvin cycle. So in the light dependent, we get water to oxygen, and we get ATP and NADH. And then from the Calvin cycle, we get sugar and NADP plus and AD plus P, which are these two are waste products. This is sugar is what we want, or glucose.
Yeah, so basically it just tells you what kind of products and
it's a
receptance is needed for each specific reactions.
I'm gonna skip the diagram on page 8 and go to 9 because 9 really is more complex.
Yeah, sure.
Okay.
Yeah, good. Let's go for now.
Okay, so we have 2. It goes the photosystem 2 and photosystem 1. The 2 goes first. 2, then 1. Really bad naming, but let's start. Okay, we're gonna get these pigment molecules. They're gonna get light. Light is gonna turn into light. What are we getting from the light? We're gonna get photons. So these photons will be converted into electrons. These photons will be converted to energy, and we'll get the electrons from the hydrogen from H2O. That's why the product of H2O is O2. O2 because we only need the H. We don't need the oxygen, so we just give it away. So the oxygen is given away for us to enjoy. But they need the hydrogen. What the hydrogen does is it donates electrons. These electrons will get powered up by the photons and go to the electron transport chain. Electron transport chain will create ATP and with the energy. So this electron transport chain, I believe, is very similar to what happens in the mitochondria. But here's what happens in the first time. Then we're going to reuse the electrons the first hydrogen gave us in the photosystem 1 with more photons. We'll power back up. We're reusing the electrons. We use the energy in number 4. Step 4, we're going to use the energy from the sun. But we're not going to get rid of the electrons. The electrons doesn't go to the ATP, but it'll go to the photosystem 1 to get recharged by more energy from the sun. And it'll go through another electron transport chain, but this time we're going to make NADPH. With the hydrogen we got. I think this hydrogen goes all the way here. The hydrogen, that's why hydrogen isn't another waste product, because we're going to use it to make NADPH. Which in the cellular respiration slides, we're going to learn that this is transporter. It transports electrons.
We kind of should have gone
maybe
cellular first. Because it explains the electron transport chain.
The problems of today cannot be fixed.
Go to the next one. I think we can kind of softly explain that.
Oh, okay. The light this is really this these green, what would you call them? Like green half circles?
You mean thyroid?
No, the one's called photosystem in 10.
Photosystem.
Like half circles?
I thought, isn't that the,
They're the photosystem 2 and photosystem 1.
it's a pigment. Isn't that the pigment?
No, this is like the entire purple thing in combined. In just made into photosystem.
Yeah,
I'm just gonna call it.
The purple.
No, I'm talking about the green.
Thyroid, right?
No, the purple are electrons, I think.
Oh, sorry. Which okay, so.
Oh, 10. I'm on 10.
Okay, so.
The green should I call them half circles with circle holes?
It's like the green circles or so.
The green half circles are representing, if we go back to 9, the purple. All the purple turns into this green half circle. So whatever happens here, we're just simplifying in this diagram to show that light hits, and here we show that 2 electrons get out, we get O2. The electrons are pushed, the hydrogen goes here. We know how we're gonna go more in depth. This is happening in the thacloid, but the hydrogen will spin ATP synthase to make ATP and NADPH. And this is gonna go to the Calvin cycle. So the light-dependent reactions make ATP and NADPH. But it also creates oxygen, which is a waste product. So now we're going to talk about the Calvin cycle, or the light-independent reaction. So this, we don't need light for this one. So the light-independent, or the Calvin cycle, please do not get this mixed up with the Krebs cycle. This is the Calvin cycle. The other one's for cellular respiration is the Krebs cycle. But we're going to use carbon fixation. Here, I think I'm going to just jump into slide 13.
For the diagram, yeah.
Yeah. And then after slide... Oh, we're almost done. Okay, so we're going to make sugar from CO2. So we need one more CO2 from the environment. We need CO2 from the environment. Because we have Ruby P. This is what we start off. Ruby P. Which is 5 carbon, 2 phosphorus. So we're going to have to make 3. We have 3 rubies. So what we want is 6 phosphoglycerate. Which is 3 carbon and 1 phosphorus. We have the phosphorus, but we don't have enough carbon. So we're going to add a carbon. We're going to split this in half and add carbon. So now we have 6, which is this in half plus 1 CO2. We're adding 1 CO2 here to make 6 and cutting that in half to make 3, 1. So now we're going to get this 6 ATP and turn, it's called reduction of this into G3P. So we're going to break ATP, we're going to break NADPH, and when we do that, we're going to get G3P. Once we get G3P, we're only going to use one. We're going to use 1, and then the 5 turns back to RUBP, and the cycle goes again. I think it says how many times? 6 times. 6 times. This process goes again, and again, and again, and again, and again. 6 times. So that's how we create sugar, and that's why we need ATP and NADPH, because we're gonna use we're gonna use this to reduce it. And what we
basically we have to break the RUP. And reform as well.
So we get 3 phosphoglycerate. That's what when we break it and add those, and then we reduce this 3 phosphoglycerate to G3P. 1 goes down, and we do that 6 times, and the 5 regenerates. This is why we need the ATP and NADPH from the light dependent reactions. So we have alternatives. This isn't very long. I'm just gonna speed through it. Types of plants. These I think are normal. These are C3 plants. They go Calvin cycle. Closes stoma during hot dry. So they'll close the stoma to conserve water. Really, I'm just gonna call one main thing. Okay, types of plants that fix carbon twice. These are C4 plants. So they also close their stomas in hot, dry days to conserve energy. But they fix carbon twice. In 2 kinds of cells, keep CO2 high. CAM cells are that fix carbon twice at different times of day. So the stoma opens at night where it's cool to conserve water instead of hot, dry days. They'll only open at night. Cactuses, the desert is hot in the day but cold at night. So that products are stored in central valve. Really, that's it. See, here's the difference. At night, there's carbon fixation. At day, the Calvin cycle. Here, this happens all at once, like in a day, and this is just Calvin cycle. We're done.